and then it's a break, and then just one more week. How did that happen? So I'm a little bit behind in what I wanted to cover this semester, but so I'm going to be tweaking things and updating things. I already updated this slide set just a little bit, and then the next one's posted, but I already realized I got to update that one too. So I'll be trying to alert you of when I'm updating the things in EduCat. But basically, I wanted to cover more on the human microbe interaction, and then the next topic is really the major last topic is bioremediation. So I'm hoping to squeeze in a little bit on that too, but it'll be a little bit truncated. And then just a reminder, the final exam is during exam week on Thursday at 10 a.m. So, okay. We left off here, I believe, looking at the idea of an ultra clean environment maybe is detrimental. And this study that when we looked at that video clip, that was one of the earliest studies that was done. This paper is, I highlighted her name, Dr. Moodius was the one that was, the documentary was covering her research between children raised on farms versus in the city and the impact on asthma. And so when her research and some other similar scientists doing, similar work, when this started to come out, this was big news. The idea that it could be driven by a lot of the exposure of the early microbes when you're young, and that basically the idea is they think that in large part that exposure to microbes in early childhood helps to work out your immune system essentially, and it keeps your immune system kind of being able to figure out between friend and foe. And so they're looking at asthma impact, allergy impact with early childhood exposure to beneficial microbes. And so they were looking at the farm exposure here again and looking at asthma rates. And so here they were tracking a couple of things that's going to keep showing up here in other things that we talk about. They were looking at butyrate levels in the feces. And so here they say the gut microbiome may contribute to asthma protection through metabolites. So they're saying it's not just the presence of the microbes, but what the microbes are doing. And that as they live their life in the gut, they're taking in nutrients, but they're also, of course, excreting their waste products, which then influence our physiology and health. So some other studies that came after that, again, looking at, okay, is there something we can do, some kind of intervention? Can we try to pinpoint this a little bit more? And again, they're supporting the idea that the exposure to microbes in early life is important. Here says, but now studies suggest that the ideal time to use microbes as preventative treatment for asthma is within, excuse me, the first 100 days of life. So they're even getting a little finer tuned here on what is a good target for the date range here. Then they even took it and kind of stepped backward in time a little bit and said, what about when the baby is still in utero? What's going on there? And so here they looked at when the mother is pregnant with the child and what impact the mother's diet and does she have asthma or other allergies? What is she eating? And could that also play a role? And so they were looking at that, and also once the baby is born, what's going on with the breast milk. And they're also showing that that can have an impact. So when you are pregnant, the things that you choose to eat and how healthy you are, they're finding now has an impact on the child in many ways, not just asthma related, but they're finding now in terms of healthy weight for the baby, allergies, things like this. It matters what the mother is doing while she's pregnant. And then this study was published just this year, looking at the idea of breastfed versus formula fed with artificial milk, and what could they see in terms of similarities and differences with that. And again, they are showing that they do see a difference with what's going on with the types of microbes showing up based on what kind of milk they're getting. And so here it says breastfed infants showed higher levels of beneficial bacteria, such as bifidobacterium and lactobacillus, while formula-fed infants had a higher prevalence of potentially pathogenic bacteria, including Clostridium difficile and Enterobacteriaceae. And so it says breastfeeding promotes a healthier and more diverse microbial ecosystem. So there's that idea again, more diverse. And so again, this is recent research going on, again, showing the impact between what kinds of, in this case, food is the infant breast milk or formula-fed milk. And so from this paper, this is an image from that paper, and they're showing, what's in the breast milk. And you certainly have immune factors. So you do have immune system cells that are in the breast milk that the baby has access to, as well as, of course, the actual nutritional profile of the breast milk in terms of the proteins and lipids and carbohydrates, the minerals, the vitamins, but also that you have immune system cells and microbes. There is a certain population of microbes in the actual breast milk itself. HMOs, that's human milk oligosaccharides, which I think they define down here in this little clip, the human milk oligosaccharides. And then short chain fatty acids, that's showing up again, SCFAs, microbiome. And so they're saying that the human milk has implications for not only, of course, the nutritional health of the baby, but helping to seed the microbiome of that infant and promoting proper growth. So this idea of, even when the infant is still developing in utero, that there is impact starting right from that point onward. And then I wanted to also point out that they're looking at this too for other diseases such as COVID-19. So they're looking at, could we have a selection of microbes that could be fed to people to help promote their ability to either not get COVID-19 or to have it less of a issue if they do come down with that. And so I was kind of shocked when COVID-19 first came out. Well, I guess I wasn't terribly shocked, but I was disappointed, I guess is more the word, that they weren't looking at this immediately. This was something that was, again, kind of fringe science. I know they're scrambling, trying to figure out what it was, how to fight it. But a lot of, I think, if they had done a little bit better job of putting it out there into the general knowledge base of general people that are being exposed to COVID-19, that there's a lot you can do to try to mitigate how severe this is by healthy eating and having a good, stable, diverse microbiome. And the studies that have come out since 2020 have verified this, that there's strong implication that people who had, good preventative health, had good diverse microbiomes, they had less issues with COVID-19 severity. And in fact, some people didn't, weren't even getting sick with COVID-19. So this is something, again, that's not, just for asthma and allergies. You can broaden this out and start thinking about it in a more universal perspective of how do we mitigate all kinds of diseases with understanding our microbiome. Okay. So that's the end of slide set one, part one. And I wanted to continue on looking at more specifically now, getting into the gut microbiome a little bit more. And that has been where most of the research is now. When you look at the number of studies published. So the National Institute of Health back in, I believe, 2005 started advertising that they were going to devote millions upon millions of dollars in grant money for people to study the human microbiome, who's there on a normal, regular basis, not the pathogens, but the ones that coexist with us through our life. And since that time, there's been tens of millions of dollars devoted to this, mostly for the gastrointestinal tract research, but the skin and oral microbiome have also had some good research being done in those parts, for those parts of the body too. So incredibly rich environment. And when you think about the gastrointestinal tract, it has, and that's conservative, 500 different species. It depends on how fine you get with your studies, all the way from the stomach on down. Helicobacter pylori has received a lot of attention for being able to live a life quite comfortably in the stomach. And the flip side is that in some cases it can cause ulcers, which was a big hullabaloo when this first came out because they didn't believe that initially. They thought ulcers was caused by spicy food and stress, and that's not really the case. I had to laugh because my dad was diagnosed with an ulcer and he was 100% Finnish. He did not eat spicy food. The Finnish population, it's meat and potatoes, pretty bland. And so, yeah, no. Stress, I could see. He did have some stressful times in his adult life, but Helicobacter pylori, a lot of us have that just naturally in our stomach, and it doesn't cause any problems at all. But for some cases, it can trigger ulcers. And they demonstrated that by one of the scientists swallowing a vial of Helicobacter pylori and saying, "Watch, I'll get an ulcer." And he did. And they gave him antibiotics and his ulcer went away. So pretty adventurous way to prove your point. I would never have done that myself, but he was very sick and tired of getting a lot of, no, you're wrong. This can't be happening. And so at a conference, he swallowed some Helicobacter pylori to prove his point. So all the way from the stomach on down, yes, there's more than just Helicobacter in the stomach. That's the most famous one. And, the reason it was such a kind of shocking discovery is that the stomach is, really acidic. They didn't think anything could handle that. But there are Helicobacter and several related species that are found in adults, not causing problems. And basically, they have a way to deal with a very acidic pH. They have a way to create a microenvironment that's not quite so acidic. And they have a way to kind of hide out in the mucus of the stomach so that they're sheltered as well. So they have some nice strategies to hang out and survive and thrive in the stomach. Then, of course, you move into the small intestine and then from there to the large intestine. And when you progress further south, so to speak, you get more and more higher and higher populations of microbes overall. And so the stomach has very low populations, the small intestine, pretty good populations, large intestine, they top out. They have lots and lots of microbes per gram of fecal matter is often how they will assess that. And a lot of them are either anaerobic or facultative anaerobes. But you can see here, they generally think that the anaerobes dominate in the large intestine, but the facultative anaerobes are also there, such as E. coli and Fusobacterium. And when you look at fecal content, about 50% of that is bacteria by weight and volume. And so with human studies, when they look at gastrointestinal tract, the only way they can do that safely is through fecal analysis. So keep that in mind. That has limitations because you can't tell exactly where those microbes in the feces lived. Did they live in the stomach, small intestine, large intestine? And so you have to take that into account when you're analyzing the study and the results and conclusions. They're working on a way to try to get more precise sampling, and I'll talk about that, but so far in human studies, that has not been possible. So the stomach. Helicobacter is just really good at surviving low pHs. There's a nice picture of it on the stomach lining, and it is an issue, especially if you have to be careful. You know, a lot of people will eat Tums or, you know, I'm trying to think of the generic name of Tums, but anyways, it's, you know, you take that, it's essentially carbonate to calm your stomach down, but it raises your pH from not being so acidic. And that can be problematic. It can also be caused by poor nutrition. So especially in third world countries, they'll see this. But here in America, people do it not really thinking about it by eating Tums and things like that, which raises the pH of their stomach. And that can have some problems. In third world countries, it often leads to cholera because of the poor water supply, the contamination of Vibrio in their water supplies. Here, we don't have that situation very often because our water supply is much cleaner. But you can create problems if you raise the pH of your stomach too much, if you happen to eat contaminated food or water, and now they don't have that acidic stomach to pass through. So they might be able to survive better and make it to your intestines and make you sick. And so this is something that they've been warning against, that you shouldn't eat things like Tums, like candy, and be a little bit thoughtful about that because it can cause problems. So here you're looking at a nice image of the intestinal cells in blue. And then the main reason I did this was to show you all the microbes are in the left corner here. So you can see a nice diversity of microbes showing up. And then the green is the mucosal lining of the intestinal tract. And so this area of the intestine has received a lot of attention in terms of research especially from a human medical perspective, because understanding this mucosal barrier and how microbes might breach that, or metabolites from the microbes might breach that. And then the layer of intestinal cells, right, you know, the very surface layer of the lumen of your intestine, how those cells are impacted by, again, the microbes or the metabolites. It's been heavily looked at, and things like leaky gut. I'm sure you've probably heard about that in just the general news media. They talk about leaky gut and irritable bowel syndrome and things like this. So this is what they're zooming in on. They're looking at this part of the intestinal tract and trying to understand this dynamic better. And so we'll talk a little bit more about that, but they are finding that you can see that there are some microbes that are in the deeper layers of the mucus. And so the thought is that microbes may be able to more directly interact with the human intestinal cells. Metabolites from the microbes are also influencing that. They've even seen shifts in gene expression in the intestinal cell due to the presence of either the microbes or their metabolites, their waste products. So lots of interest in this area. Huh. I don't know why it does that. Let's see if I can go back here. Okay, I don't know why it's doing that. Get it to erase. So if you've not thought about your intestinal tract, here it is. And so I'm not going to ask you to know all the subparts of the intestine, but understanding stomach, small intestine, large intestine, and understanding that you get increases of pH as you move your way down. And getting more toward neutral by the time you reach the colon. Again, this is a very cursory listing of microbes. There's way more than this, but there's trying to show some dominant microbes. And then also, I guess it's kind of, they're trying to point out that there's more microbes in the large intestine simply by how much more they are listing out there. But I think this next slide gets a little bit more into some numbers where you have showing the per mil of fluid in the stomach, small intestine, they switch to grams in terms of what's there in the fecal matter. So these are some of the studies coming out. Now, if they actually are testing the small intestine and they talk about it per gram of small intestine contents, you know that they are talking about mice or rat studies because they don't do that to humans. So if they are actually getting this kind of data where they know, there's up to 10 to the 7th per gram in the small intestine, that's based on mouse studies. So just keep that in mind. But the assumption is generally what we see happening in mice is analogous in humans in many cases, but not always. Here again, just to get you to think a little bit about your intestinal cells rate in the area. You have to imagine all the food and bacteria are in the lumen of your intestines. And then the surface of your intestinal cells are very convoluted like this. And so you have great surface area for absorption of nutrients. And then that gets passed into your bloodstream. So when you look at it here, geez, I wish I could get rid of that. It won't even let me. There's no way to erase it. Here they're doing a nice idea here. If you've not thought about, what's going on in your intestinal tract, they're trying to show kind of the step-by-step process with the digestion process. And it's not really till you get to the small intestine that you get some really good transfer of the nutrients into the blood. And then, of course, the fecal matter at the very end. So this idea, there's a paper published on a study where they looked at this idea of the gastrointestinal tract all the way from the mouth to the rectum and looked at, it's kind of like stream ecology. So for those of us who have taken classes like this, and since we're biologists, I thought this was a good analogy to think about. I've posted this paper in EDUCAT, so you can find this information there for study purposes. But basically, they were looking at, when you think about it, when people eat, that's kind of like the headwater of a river where you have the start of it all, and then outflow into the ocean is like at the other end of the gastrointestinal tract, out the anus. And so, they said, well, let's look at a stream and think of the upper river region. You have typically pretty high water velocity and pretty good oxygenation because of that water movement. And that's similar to what we see in the small intestine, where you do have faster movement and you have more microbes that are either facultative or even aerobic in the small intestine. And then as you get into the middle region of the river tends to slow down and you get some nice species richness. So better numbers of microbes. And that's what we see in the small intestine as well. And leading into the large intestine, you get slower movement of materials through that portion of the gastrointestinal tract. But you have lots of microbes there, lots of different species. And then as you work your way further down the river, that's where it tends to be the slowest moving and a high sedimentation rate, just like with fecal matter starting to build up, and low oxygen and lower diversity right at the very end there. And that's what they see in pretty much with the large intestine too, especially right at the very end. So a nice analogy, if that helps you think a little bit more specifically about the process of the gastrointestinal tract. I thought that was a helpful way of looking at it. They went into it in a little bit more detail. They said, if you think about the riparian zone, which is the area of like the banks of the river, if you want to think of it that way, that's the riparian zone. And they said, that's where you have nice exchange, the banks of the river, you have nice exchange of nutrients between the river and the land because of that, the soil and the matrix of the land right there. And they said that's very analogous to the mucosa of the intestine. When you look at the lumen and the surface intestinal cells, that's like the riparian zone because that's where you get exchange of nutrients between the gastrointestinal tract and the host. So it's very similar. But in this case, they said definitely the microbes really do influence the host much more strongly than microbes do in the river. So they, definitely have that. And so looking at the idea that the microbes that hang out right against or in the mucus and right near the human intestinal cells, they even influence the development of those intestinal cells. So the actual physical nature of those cells and the way they got into this study was through mice and rats, and they were looking at if you keep mice in a germ-free environment, what would happen to their intestinal tract? And they could show big differences. The intestinal tract was shorter. The intestinal cells, especially the lining of the cells, were different physically. And the gene expression in those cells was different, whether it was germ-free mice being raised in a germ-free environment or those colonized by their normal, just standard microbiota that are in mice colonies. And so definite difference too between which microbes tended to be on and in the mucus versus the microbes that are more in the central portion of the intestine. So again, this would be analogous to microbes that are right up against the bank. They're part of the river, but they're right there against the surface of the bank of the river versus the microbes that are in the midstream flow of the river. Definite differences. And then the study, what they thought about too, and was looking at through some research, they said, what would happen if the ecosystem had a major disturbance? And what they were looking at from the human perspective was food choices of the host and what happens if the host experiences diarrhea. And so thinking about that kind of a severe disturbance. And as you know, when you go into an ecosystem and disturb it, we've already been talking about how that can really strongly influence the microbial populations, the diversity, and the successional processes of those microbes. So this is the paper that I'd like you to read. And they looked at the effect of diarrhea on the gastrointestinal ecosystem and how fermentable fibers can be used as a management tool. You often hear that in ecology studies, a management tool to promote health of the gastrointestinal tract and recovery from diarrhea. So fermentable fibers, by the way, are prebiotics. Essentially, fermentable fibers are prebiotics. So it's stuff that microbes in your gastrointestinal tract use for their nutrition. So please read that and see how the story ends with the diarrhea event and what they're suggesting. So I thought that was a good analogy to get you thinking about your intestinal tract in a slightly different way. So they've known this for a long time, even before millions of dollars were being spent on more sophisticated research. They've known for a long time that microbes that exist on and in you, the beneficial ones, one reason we say they're beneficial is that they provide this service to you, preemptive colonization. And so if you have a healthy microbiome in your intestinal tract and you eat something that's contaminated, drink something, there's probably been many cases in your life already where that has happened to you, but because you had a beneficial microbiome there, you didn't get sick, or maybe just slightly sick. And then there's the case where if you disturb your microbiota, where you wipe out a lot of them, and then eat that contaminated food or water, you're at much higher risk of getting seriously ill. So preemptive colonization, same with your skin too. The amount of times that you have interacted with potential pathogens, with getting them on your skin, on your hands. And then, there's, again, chances are you've had that happen to you over and over and you just didn't know it because you had enough microbes on your skin that were already there colonizing that space where the pathogen couldn't get any foothold, so to speak. And so they've known about this for quite some time. And then also this too, the idea that microbes in your gastrointestinal tract, what you eat feeds them as well. And so they're able to extract nutrients from the food that you eat. And often it's, nutrients that you yourself don't break down because you don't have the enzymes for it, but the microbes do. And so they'll use some of that for their own energy needs, but they can also release some of that back to you in forms that you can use and make use of it as part of your nutrition. So this is an older picture showing the intestinal tract and how some of the microbes are right there on the surface in the mucus area. And so they've known, this is an older table, they've known about this idea for a long time. Even steroids that you have, some of the steroids can't be fully made without the help of certain microbes there. And so your steroids don't mature, some of them, until the microbes have acted on them. When you were first born, you probably got your very first shot was a vitamin K shot, most likely. I think they still do that. So they know that E. coli in particular produces vitamin K for you. But when you're born, you don't have any E. coli in you yet. And they recognize that babies should have that exposure right away, especially in this day and age. And so they will usually inject you with a vitamin K shot. So it's bad enough you have to go through the whole being born process, but then they stick you with a needle right after that. So gas production, certainly, we know that many microbes produce all kinds of gases. And so this is an older table. So this is something that they've known about for quite a while. And then as the research has gotten more sophisticated, they're finding out more details. And so they're even showing that the presence of certain microbes can really influence what your intestinal cells are doing. In this case, this particular study, they were showing how a Bacteroides species was able to interact with the intestinal cells and get it to produce a protein to kill other bacteria. So kind of bizarre interactions here that they never knew, in the older research. They just didn't have the technology to get into this. And so again, the idea that with germ-free mice studies, big difference in the actual length of the intestinal tract, the morphology, even the amount of, when I was showing you the picture of the villi, how lovely that was with the invaginations like that and that great surface area. They even show that that kind of gets flattened out in germ-free mice. And so you don't have that great surface area in germ-free mice. The presence of the microbes help mature your blood vessel development in that area as well, which is super important, of course, because you have to have really good blood vessel in that area for, of course, absorption of nutrients. And so the fancy word for that is angiogenesis. And we'll talk a little bit more about this, but the majority of your immune system is actually in your gut. And so this idea that if you have germ-free mice and look at the development of the immune system of those mice compared to the mice that were raised conventionally with the normal microbes, huge differences in their immune system maturation and function. And dysfunction is probably a better way to say it. And then, like I said, some of the more sophisticated studies are definitely showing that the microbes are influencing your gene expression of your eukaryotic cells in your intestinal tract. And so that's a pretty intimate interaction there, where it's actually influencing at the DNA level what some of your cells are doing. So much more interaction than they first thought. And what really tended to kind of break open this field of study was this idea of germ-free mice, looking at germ-free mice versus conventional mice and comparing the similarities and differences. And that's when they really started to get some pretty exciting data showing some major differences. And so a lot of the research is showing that you need a healthy intestinal tract lining, the mucus, the cells right there need to be healthy. We'll talk about tight junctions and all of that in terms of leaky gut. And they say, and you need that so that the microbes stay where they belong, which is in the intestinal tract. It's fine if they're, interacting as long as they're staying in the intestinal tract, especially when you think about some of the pathogens, of course. And they're finding more than one study has definitely been showing that even emotional stress can impact your microbiome. Certainly, your diet choices will do that too. We've talked about antibiotic therapy already more than once, how that can strongly influence your microbiome. And so when we already talked about this idea of dysbiosis, how it's an imbalance of the microbes, their populations and ratios, and that they see with that imbalance all sorts of negative impacts. And one of the biggest diseases they're studying is inflammatory bowel disease, because that's been such a mystery of why some people get this and the related diseases to this. So again, it's pointing to this idea of there's some kind of dysbiosis, and we need to understand that better. So some of the earlier studies, as I said, were looking at germ-free mice and compared to the normal or conventionally raised mice. And this lab, they were looking at the idea of what's contributing to obesity in the human population. It's actually referred to as an epidemic, and some people say pandemic, worldwide issue, because of the rates of obesity have gone up. And so they started to look at this through these mouse studies where they had germ-free versus conventionally raised. And what they found, so germ-free are the white boxes, and conventional, they had two different treatments of conventional. So the big take-home picture here is that you look at the percent body fat by the end of the experiment, the mice that were germ-free were more lean, they were skinnier. But if you compare that to how much they ate, they actually ate more food. So the mouse food that they were provided, they actually ate more, but gained less weight. In fact, they didn't gain weight. Whereas the conventional, you could see they weighed more at the end of the experiment, but they ate less. So this was a head scratcher. And like, wait, what? So basically what this is saying is that something's going on with the microbiome that's influencing this. Because if you have no microbiome, they don't weigh as much, even though they ate more. So it must be something to do with the microbiome. So then they started looking at the microbiome conventionally raised and started to look more carefully. And basically what the study was showing is that it's really how well the microbiome was extracting energy from that food. And so if there's no microbes there to extract energy from the food, then those mice didn't weigh as much. They were skinny at the end of the experiment. But if the microbes are there, they're eating some of the food you're eating, they're using some of it, but what they don't use, they give back to you. And so they're providing some caloric energy back to you, which allowed the conventional mice to have the higher percent of body fat at the end of the experiment. And so the obese microbiome has an increased capacity to harvest energy from the diet. That was a huge take-home message right there. Then they said, furthermore, this trait is transmissible. This was another interesting part of their experiment. Colonization of germ-free mice with an obese microbiota resulted in a significantly greater increase in total body fat. So they were able to transfer the trait of obesity by transferring the microbiome from obese rats. Pretty amazing. When this study came out, this is a 2006, this was like, I remember NBC News was carrying this. They were like, everyone was just like, what? It was very exciting data. And so they're showing that what microbes are there and how well do they extract energy can influence the host's body weight. And so this was very exciting because then they said, oh, well, then if we could understand that better, then maybe we could come up with treatments where we can give people a mixture of microbes that would essentially regulate the host's weight in a better way and not cause obesity. And so kind of like an anti-obesity probiotic pill is what they're envisioning. And that, you know, someday they'll be able to provide really precise medical advice to people that are struggling with obesity by manipulating the microbiome, which is a pretty amazing angle of attack of ways to try to attack this this issue, this medical problem. And like I said, so many people are struggling with obesity that can, and it's very, this isn't the only thing leading to obesity. This, you know, dysbiosis, I'm not saying that. There's many different things that influence it, but this is one aspect of it that they're very excited about because they said that, you know, this can be used as a therapeutic target. And so if we can understand this a little bit better, we could potentially detect it earlier in people that are, you know, on the path to obesity, but not there yet, by assessing their microbiome and then do like an intervention, an early intervention, so that person never does develop obesity and all the associated diseases that could happen. And then a therapeutic target too, to help people that are struggling. And so this is, you know, lots of scientists and medical professionals are very, very excited about this because they see that this could really help add to their arsenal of how to help people and how to prevent it to begin with. And so the more they look at the microbiome, especially in the intestinal tract, the more they're seeing how pivotal they can be for the host's health. This is taken from a paper. And this is just trying to show some ideas that, certainly the maturation of the immune system, it helps protect against pathogens. It helps with the maturation of the epithelial cells of the intestinal tract. Of course, it helps you digest your food. All right, so the more they look, the more they're finding. And so we're going to get into this a little bit more and look at what else these microbes are doing in your intestinal tract, but I see we're out of time, so we'll do that on Wednesday.